Myself, Dr. Raktim Mukherjee, Assistant Professor and Head of the Department, Shri P.M. Patel Institute of PG Studies and Research in Science. The institute is affiliated to Sardar Patel University, Anand, accredited with NAC and recognized by UGC 2F and 12B. The title of my today's presentation is Structure of MHC. In this lecture, I shall describe the structure of MHC class 1 followed by structure of MHC class 2, and thereafter I shall compare the two structures based on various points. So let's begin with a fantastic cuisine. In fact, the top panel, if you see, the top panel is a classical Gujarati thali that is being depicted. Whereas the bottom panel depicts a small kid with a huge chunk of watermelon. So what I am trying to convey from these two slides based on the structure of MHC. The point I would like to convey over here is that although this Gujarati thali or Gujarati cuisine consists of so many menus, say Shrikhand, the Kopra Paks, the various veggies, the saves, but you cannot have the entire thali at a single go. You may have one menu at a time. Similarly, the child is holding a huge watermelon, but the child won't be in a position to eat this entire watermelon in a single go. But the same, because the child's mouth or oral cavity is limited space. But the same watermelon, if you cut or chop into small pieces, the child can gobble each piece separately. So this is what I am trying to convey. Now let's come back to my context of major histocompatibility complex. Each MHC molecule has one binding site, just like the child has one entry point for eating, that is mouth. Similarly, the MHC molecule has one binding site. There are different peptides that bind to the same site. When I say different peptides, I refer to different food can enter the mouth, that is the same site is the mouth, but of a given MHC molecule, but only one at a time. You can have all the food stuff, but one at a time, not that the entire food can be gobbled down at a single instance. Further, when I go to the structure of class 1 and class 2 MHC molecule, first I shall focus on class 1, and after I complete class 1, there I go to class 2 structure. So let's focus on MHC class 1. This is the general structure of MHC class 1. Before I go into the details, let me brief you that it consists of alpha 1, alpha 2, alpha 3 and beta 2 microglobulin fragments. So what I will do is I'll write over here, okay, I'll write somewhere here alpha 1, alpha 2, here you have alpha 3, and finally you have beta 2 microglobulin. So let's focus on the first three fragments, the alpha 1, alpha 2 and alpha 3, the first three fragments. So there it is. I am focusing on the first three fragments. These three fragments together, that is alpha 1, alpha 2 and alpha 3, they comprise of a 45 kilodalton fragment, alpha 1, alpha 2 and alpha 3. And then this alpha 1, alpha 2, alpha 3, this 45 kilodalton fragment consists of three external domains, alpha 1, alpha 2 and alpha 3, and each of these domains is having 90 amino acid. So it has three domains, alpha 1, alpha 2 and alpha 3, and each of this amino acid is having 90 amino acids. Each domain has 90 amino acids each. Now let's focus on the missing fragment. The missing fragment or the one which comes later is this 12 kilodalton fragment. This 12 kilodalton fragment is known as beta 2 microglobulin, and this beta 2 microglobulin fragment is very similar to the alpha 3 domain or this alpha 3 fragment. Next we have the membrane distal domains. Distal is towards the top. The membrane distal domain consists of alpha 1 and alpha 2 fragments. Between this alpha 1 and alpha 2 you can see a cliff, the valley or a ridge, and this ridge or valley is your peptide binding cleft. This is precisely the site where the peptide of class 1 MHC will bind. This is the membrane distal. So someone asks you in exam which are the components of membrane distal, the answer should be alpha 1, alpha 2. Thereafter you have membrane proximal domains. When we talk about membrane proximal domains, the membrane proximal domain consists of alpha 3 and a class 1 glycoprotein which is known as beta 2 microglobulin that we saw. So membrane proximal domain consists of alpha 3 and beta 2 microglobulin. Remember students that beta 2 microglobulin is very similar to alpha 3. Next we have a transmembrane segment. The transmembrane segment consists of 25 hydrophobic amino acid and a short stretch of hydrophilic amino acid, and finally a cytosolic tail. The cytosolic tail consists of hydrophilic amino acids of 30 anchor segment hydrophilic amino acid. When I refer to the terms hydrophilic and hydrophobic, hydrophilic is water loving and hydrophobic is water hating or water repulsive. This can better be explained with an analogous example of Lord Hanumanji. You might have heard about the story that when Hanumanji was taken to Lanka, Ravana did a big mistake because he could not understand the prowess of this great monkey. He thought Hanumanji as an ordinary monkey, and what he did, the biggest mistake, he doused the flame of Lord Hanuman. And the rest of the story is known to you very well, what happened to Lanka after that. Hanumanji set himself free. He jumped all across the Lanka and destroyed each and every property, leaving only two places intact. One was the place of Vibhishana and another was Mandotari, because these two were religious person. But I am not going into the details of the story. Instead, my focus is that after Hanumanji's tail was doused, it is said, according to mythology, he went to the ocean immerse the tail to douse off the flames. So my dear students, the bottom part of the tail, the bushy region which was set on fire, that you can compare with this cytoplasmic tail or hydrophilic amino anchor segment of 30 amino acid. Rest of the portion, the hydrophobic amino acid, a short stretch of hydrophilic amino acid. Okay, so that part which goes initially into water helps you to remember that the hydrophilic anchor segment is of 30 amino acid. Moving ahead with the talk, when the same MHC was dissected with an enzyme PAPin, the PAPin would fragment the enzyme, and after fragmentation, what it was found, you all purchase Amazon and Flipkart boxes. What you see, the bottom of the boxes is very profound. It's very nicely managed with lots of hard brown colored sellotape being pasted so that the contents doesn't come out. So similarly, the bottom of the box is very sturdy. It's the beta strands of beta plated sheet, whereas the sides are made up of alpha helices. Obviously the content, if you have to take it out, it will be from the top of the box, and the top of the box is made up of peptide binding cleft, and this peptide binding cleft is, in case of class 1, is 10 amino acid. In fact, the range is 8 to 10 amino acids, and the optimum size is 10 amino acids. The sides are alpha helices. Then you have the beta strands of beta plated sheet towards the bottom. So that is the structure of MHC after it has been digested using the enzyme PAPin. Remember students, PAPin enzyme is derived from raw papaya as a common source. Now after we covered or completed class 1, let's focus on class 2 MHC molecule. So my next focus is class 2. When you talk about class 2 MHC molecule, the membrane proximal consists of alpha 2 and beta 2 domains, and the membrane proximal, like the membrane proximal alpha 3 and beta 2 microglobulin of class 1. So the microglobulin domains of class 1 MHC molecule be a sequence like immunoglobulin fold structure, and this is the reason MHC class 2 molecule are also classified in the immunoglobulin superfamily of receptors. So let's study class 2. Class 2 MHC molecule consists of a 33 kilodalton fragment consisting of alpha 1 and alpha 2, 33 kilodalton fragment, and a 28 kilodalton fragment consisting of beta 1 and beta 2, a 28 kilodalton fragment. Just like the previous case, between alpha 1 and beta 1 you have the peptide binding cleft. So that's the difference. In case of class 1 it was alpha 1 alpha 2. In case of class 2 it is alpha 1 beta 1, which makes the peptide binding cleft. Likewise, rest of the areas are similar. You have a transmembrane segment. This is very similar to MHC class 1, 25 hydrophobic and a short stretch of hydrophilic amino acid, and a cytoplasmic tail consisting of 30 amino acid anchor residues. So there you have the membrane distal domain. In case of MHC class 1, it's made up of alpha 1 and beta 1, membrane proximal domain made up of alpha 2 and beta 2 fragment, the transmembrane segment and a cytoplasmic tail. So the transmembrane segment and cytoplasmic tail is very similar to that of MHC class 1. So I'm not going into the detailed discussion. Now when you look from the top, the cleft geometry of class 1 and class 2 MHC molecule, especially the peptide, you can see that MHC class 1 can accommodate peptides of 8 to 10 amino acid, whereas MHC class 2 can accommodate 13 to 18 amino acids. So in general words, always it is said that the MHC class 2 is also a structure, is also called as hot dog over the bun, because hot dog over the bun, why it is called as the hot dog over the bun is, the bun is of limited diameter. I will just draw over here, this is your bun, is of limited diameter. But if you want to place a hot dog beyond the bun, above the bun, the hot dog, the sides of the hot dog will dangle out. Similarly, you can see this is the size that can be accommodated. These portions of class 2 dangle out, and that is the reason class 2 can accommodate peptides of 13 to 18 amino acids. This is more than 13 because it can dangle out, whereas the peptide length in class 1 is fixed, 8 to 10 amino acid. And what is the reason? The best reason is, can be analogous to this, your cloth is hanging on the string, there is anchor residues over here and the anchor residues over here, no support in between. So whenever you are giving wet clothes, the string which was initially at the top would now dangle down as more and more weight of cloth comes. Another example which while drying clothes is in an apartment. In apartment what happens? You have a huge string placed end to end, but the supports or the anchors are not just towards the end. You have a pole, right, at regular intervals, say every 1 meter. So irrespective of whatever weight of cloth you give, the string simply won't bend. The string won't bend. So this is the advantage. So one is having anchor residues only at both ends, and another anchor residues are not only at the both ends but also at regular intervals to keep this upright, straight, or at constant elevation. I would use the word constant elevation. This is constant elevation, and here it dangles down, not constant elevation. Based on this, let me end my lecture with this comparison between peptide binding of class 1 and class 2 MHC molecule. Peptide binding domain in class 1 is alpha 1, alpha 2. Peptide binding in class 2 is alpha 1, beta 1. Already we have seen. Nature of peptide binding cleft. In case of class 1, it is closed at both ends. In case of class 2, it's open at both ends. And this is the reason we said that it can dangle out in case of class 2. General size of the bound molecule. In case of class 1, it is 8 to 10 amino acid. Class 2 is 13 to 18 amino acid. Peptide motifs involved in binding MHC molecule. There's an anchor residues at both ends, as I said. Generally the hydrophobic carboxyl terminus anchor, and here the anchor residues are distributed along the length of the peptide, and this I was explaining with my string example that it is distributed along the length of the peptide. Nature of the bound peptide is an extended structure in which both ends interact with MHC cleft by the middle arches up away from the MHC molecule. So constant, the middle one, because the string can go in up and down direction, the middle will arch up. This is MHC class 1. Whereas in MHC class 2 is an extended structure that is held at constant elevation. This figure, that unlike arching up or down, this is held at constant elevation from the floor of the MHC cleft. So that's the basic difference between MHC class 1 and class 2 molecule, based on the peptide binding domain, the nature of peptide binding cleft, the general size of bound peptides, the peptide motifs involved in binding to MHC molecule, and the nature of the bound peptide.